Most people treating brain fog as a sleep problem or a stress problem are misdiagnosing it. The actual mechanism, in a lot of cases, is metabolic. Visceral fat is running a continuous tax on prefrontal cortex efficiency, and it's doing it through a well-documented inflammatory pathway that most performance-focused people have never looked at. We're going to cover what visceral fat is actually secreting, how that signal reaches the brain, and what the intervention data shows in terms of real cognitive effect sizes. What visceral fat is actually doing, that subcutaneous fat isn't. The first thing to get clear on is that visceral adipose tissue is not passive storage. It's metabolically active tissue that continuously secretes pro-inflammatory cytokines, primarily IL6, TNF-alpha, and CRP. This is a constant low-grade output, not a response to injury or infection. It's running in the background regardless of what you're doing. Now, here's where the BMI framing breaks down. Two people can have an identical BMI and a completely different inflammatory profile depending on where that fat is distributed. Subcutaneous fat, the fat just under the skin, doesn't produce anything close to the same cytokine output as visceral fat does. So the number on the scale, or even your total body fat percentage, doesn't tell you what you actually need to know. Where it sits is the variable that matters for brain risk. The cohort data on this is consistent across multiple large studies. Anand and colleagues, Chang and colleagues, and Morris and colleagues all found higher visceral fat linked to lower scores on processing speed and executive function tests, and that relationship held up even after adjusting for vascular risk factors and other confounders. This isn't a BMI story or a cardiovascular story. Visceral fat carries its own independent cognitive signal. The effect appears across the lifespan. Schwartz and colleagues found it in adolescence. Strauss and colleagues confirmed it in overweight and obese adolescent populations specifically. And the effects tend to be stronger in females, which points to hormonal and body composition differences in how VAT is distributed and how it interacts with inflammatory pathways. There's also PET imaging data that sharpens the picture. You and colleagues used FDG PET to look at visceral fat metabolism in patients with cognitive impairment and found that higher VAT metabolic activity correlated with lower cerebral glucose metabolism in the exact cortical regions that go first in Alzheimer's. Regions tied to executive function and language processing. The brain isn't getting the fuel it needs, and visceral fat metabolism appears to be part of why. The frame to take away from this section. It's not about weight. It's about what a specific tissue depot is secreting into your bloodstream every day, and the downstream effects of that on how your brain functions under cognitive load. The inflammation to prefrontal pipeline. So the inflammatory signal is real and it's continuous. The question is how it gets from the abdomen into the prefrontal cortex, and what it does when it arrives. The clearest human evidence comes from a mediation study by Canaval and colleagues, looking at adults with excess fat mass. They measured visceral fat, plasma CRP and IL6, and then assessed executive function using a flanker task, a task that specifically demands inhibitory control and selective attention. What they found wasn't just a correlation. CRP statistically mediated the relationship between VAT and accuracy on incongruent trials. IL6 statistically mediated the relationship between VAT and P3 peak latency. That latency gap was tens of milliseconds, measurable slowing in attentional processing speed, explained by the inflammatory pathway, not by some other confounding variable. The routes by which peripheral cytokines reach the brain are well characterized. They cross the blood-brain barrier through leaky regions and specific transporters. They signal centrally via the vagus nerve. They activate endothelial cells, which produce secondary inflammatory mediators that propagate the signal inward. Once inside, you get microglial activation. The brain's resident immune cells shift into a pro-inflammatory state and start releasing their own cytokines. Neurovascular coupling degrades. The brain's ability to ramp up blood flow to active regions during cognitive demand gets blunted. Studies using functional near-infrared spectroscopy show that higher adiposity is associated with weaker task-related increases in prefrontal oxygenation. The brain can't meet the metabolic demand of a hard cognitive task the way a lower inflammation brain can. Dopamine synthesis takes a hit too. IL6 and TNF-alpha deplete the cofactors needed for dopamine production in the prefrontal and striatal circuits that govern motivation, effort allocation, and decision-making. Glutamate signaling gets disrupted. The result is a cortical network that's noisier, less efficient, and slower to reach decision thresholds. Computational modeling captures this cleanly. When researchers apply drift diffusion models to inflammation data, they consistently find two effects: a lower drift rate, meaning the brain accumulates evidence more slowly, and a higher boundary separation, meaning it requires more evidence before committing to a decision. Both of those changes increase decision latency. You're not just feeling foggy. Your decision architecture is running a measurably slower process. The cardia cohort puts a number on the long-term version of this. Participants with persistently elevated CRP from their 20s onward scored roughly 0.2 to 0.3 standard deviations worse on executive function tests at midlife, compared to the low inflammation group. That's not a dramatic effect in isolation, but it's accumulated over decades of chronic low-grade inflammation driven, in large part, by visceral fat. The cognitive cost compounds the same way the inflammatory signal does: quietly, continuously, and without any obvious acute event to blame it on. What the intervention data actually shows: the mechanism is well supported, but the practical question is whether reducing visceral fat actually moves the cognitive needle, and by how much. On the inflammatory side, the numbers are concrete. A 10 to 20% reduction in visceral fat typically produces a 20 to 30% drop in CRP and a 10 to 25% drop in IL6. Those aren't marginal shifts. They're meaningful changes to the same cytokine levels that the mediation studies show are driving attentional slowing and reduced decision accuracy. The cognitive effect sizes that follow are in the small to moderate range, roughly 0.2 to 0.4 standard deviations of improvement in attention, inhibitory control, and processing speed across weight loss and lifestyle intervention studies. That's consistent with what you'd predict from the inflammation reductions, and it's consistent across different populations and measurement approaches. Small to moderate at the individual level still translates to meaningfully faster, more accurate performance on the kinds of sustained, high-demand tasks that executive work actually requires. Brain structure data makes the case more sharply. 2 weight loss trials tracked brain PAD, a measure of how structurally old the brain looks relative to a person's chronological age. The short-term trial, 4 months in postmenopausal women, showed a statistically significant reduction in brain PAD. The long-term trial, running nearly 40 months with weight loss, maintenance, and regain phases, showed a partial eta squared of approximately 0.38, a large effect by any standard. The brain was structurally younger in sustained reduction conditions. Insulin resistance and leptin tracked with brain PAD across both trials, reinforcing the metabolic inflammatory axis as the operative mechanism. The mediation fraction matters here too. CRP and IL6 account for roughly 15 to 40% of the cognitive benefit observed after VAT loss, depending on the outcome measured. That means inflammation is a genuine pathway, not just a correlate that happens to move in the same direction. The remainder of the benefit likely runs through improved insulin sensitivity, better vascular function, and reduced cerebral small vessel burden, all of which are also downstream of visceral fat reduction. On interventions specifically, aerobic exercise is the most reliably VAT-targeted tool in the literature. It preferentially reduces visceral fat over subcutaneous fat, and it lowers CRP and IL6 through mechanisms that are partly independent of fat loss itself. Mediterranean-style dietary patterns reduce inflammatory markers alongside VAT, working on the same cytokine axis. These aren't separate strategies. They compound on the same biological pathway. For monitoring, BMI isn't the right instrument. The dashboard that actually reflects what's happening metabolically is HSCRP, Illinois, 6, and fasting insulin or HOMA-IR. Those markers tell you what the tissue is doing, not just how much of it there is. One honest gap: no large randomized controlled trial has yet directly measured prefrontal oxygenation before and after a VAT-targeted intervention, with inflammation as a formal mediator. The causal chain is plausible, well supported by converging evidence, and consistent with everything the mechanism predicts. But that specific experiment hasn't been run at scale in humans yet. The pathway is solid. The final link is still inferential. For anyone optimizing for decision quality under pressure, that gap doesn't change the practical position. Your metabolic inflammatory status is a performance variable. It's measurable, it responds to intervention, and the data shows it tracks with the cognitive output you actually depend on. Visceral fat isn't a cosmetic problem. It's a chronic drag on the same prefrontal hardware you use to make decisions under pressure, and the inflammatory pathway connecting them is real and quantifiable. If this kind of evidence-based breakdown is useful to you, Subscribe and check out the next report in the series.